have, yeah, there's up, we might as well. I don't even got any scheduled for who's gonna go first or anything. I had a couple things that I did want to talk with you guys about before we got started. Let's start with the bad news first. It shouldn't really affect us too much for like what to expect from the final and everything. But I was notified by somebody that someone in one of my past classes or current class, I don't know who it is, but somebody took, you know how I open up you guys' exams for you guys to review, like unit 1 and 2 and 3, and you guys can see your right answers and what you got wrong and help you study or whatever. And I figure most people are adult about it. And you're not, it's part of the academic honesty policy that we have in place at OCOKC. You're not allowed to disseminate that in any way. I don't know if you guys know that, but any of the materials that I give you guys, if you disseminate it outside of the campus, make it available on YouTube or something like that, that's considered academic dishonesty. And to take a test with all of the questions, the right answers, and make a YouTube video of it and disseminate it to my students, if I find out who did that, I will have them expelled. I will die getting them expelled. So if I find out who that is. So if you happen to know who may have done that or something similar, tell them to remove it immediately, because that is not just me being pissy. That is probably not just happened with my stuff, but other professor stuff is too as well, if that happened to me. So they want to get rid of that, get rid of the evidence, save their asses, because even if they're already in the nursing program, they'll get kicked out and they'll have to start from scratch. And good luck with an expulsion on your record getting into another university and starting over. I will bring you down if you do that. So I'm trying to make it easier for my students who are trying to do well in my class. So if you know somebody who is doing stuff like that, please let them know, or if it's you, take it down now. I'm not going to be like hardcore pushing on it or something, but if it comes across to me that I find out that somebody did do that and who it is, then I will bring it to the disciplinary board. So keep that in mind. So going forward, typically on my final exam, and I even will give you guys a little bit of a heads up about it, that you're going to present today, right, Angie? I guess, yes. Okay, I just want to be sure that I got that. Yes, okay. Oh, it's a lot today. It's a lot lately. Anyways, so I'm stuck on it. Just don't do that. It's part of the policy. I know people don't really, read through it. You kind of assume that you are a good person and you're not doing stuff like that. You don't have to worry about it. But if it's you, take it down. I'm not actively pursuing it. How it affects you guys is typically on my final exams, I will include questions from the past exams, because you guys did work on them and you did well on them and you're studying on them or whatever. But now I'm going to be rewording all of those questions. They're not going to be made harder or anything, but I don't want them to be the same as the questions on the past exam. So just be aware that if you are reviewing the past exams, that you understand why the right answer is the right answer and why the wrong ones are wrong, so that when you see them again, you'll see the content again. It just won't be the same exact question, because I cannot abide by that. You guys have been working way too hard to pass this class, and I can't have dicks like that ruin it for everybody and people that think that that's okay. The last time I checked, the whole reason is that you guys are trying to get into nursing or vet or wherever it is you're trying to go next. And you're trying to do that in a way that hopefully you will actually be effective at those jobs, and not just like memorizing a question and an answer and not being able to use that knowledge later on. That's why they have you go through the classes. So just looking at a question and then just knowing the words and knowing the word to pick isn't that. And that's why I run the class the way that I do. So anyways, so there's that. So if you know somebody that is involved in something like that, just give them a heads up. Like I said, I'm not like creating like a task force or something. Don't make me create a task force, basically. So if this comes up again in any other sort of situation, then that might be a thing in the future. God, hope not, guys. My plate is full enough. So, but yeah, so if it's happened to me, it's probably happened to other professors. I've told the other professors about it, and I don't want to have to bring it up to administration. So just let's not do that anymore. If you are somebody who has found a video like that has my questions and answers on, I'm not asking necessarily to bring it to me, but just tell the person, if you know them, that they shouldn't be doing that. And if you find that, I would stay away from it if I were you, just to save your own ass. So you don't get caught up in that as well. Not that you would necessarily get expelled for using it, but you could get disciplinary action in other forms. So especially for not reporting it, like that's part of those, like part of the policy as well. You're supposed to report that stuff. Not my class, but other classes where like Dean in his class, he had people like that'll just pull out like their phones to like record his lectures while he's lecturing and stuff like that. And you guys take pictures and stuff. I'm not too bothered by it. But there is a policy in place here on campus that you have to get permission from the instructor to record anything. And you're not allowed to disseminate it online at all, ever. So even like, I can't give you the permission to do that because it belongs to OSU OKC. It doesn't belong to you. I even like putting it on YouTube and stuff is like borderline me doing it is not necessarily something they like too much. So it's considered their property. So just keep an eye on that too. That's not a big deal, but going forward in other classes, maybe keep that in mind. So last last night I was like, I got home at midnight from setting up the lab and was getting in my little bath and all of this and stressed out from everything. And I just couldn't sleep last night. And I don't know if you guys have ever heard of these things called hypnagogic or hypnopompic hallucinations, but you probably experienced them and just didn't know that's what it was. And what it is like when your brain creates dreams, and how you can go through a dream and it's like real when you're in the dream, but then you wake up and you know it was a dream, right? So that part of your brain can work when you're not quite asleep. So when you're falling asleep or when you're just falling asleep or just waking up, you're like in that's called hypnopompic or hypnagogic, depending on if it's before or after the sleep cycle. And your brain will be doing dream shit while you're not quite asleep. And so you can see dream stuff. It's proper hallucinations, like where you would be like, it's not crazy hallucinations, but it's just part of the dream process, where you'll like see things like spiders on the wall or people standing in the corner of your room. Some people think like the Hat Man or like the Hag, that sort of stuff. It's like pretty typical stuff. Those are usually, if you can still move while you see that, hypnopompic or hypnagogic hallucinations. So it's very common actually, but I had hypnagogic hallucinations last night about the Russian coming into my room. And they were coming in, like soldiers were like standing in my room, and I kept like having to turn on the lights, and then there would be nothing there. But like those like 3 times, not even joking, that something like that happened. There was the Russians, and then there was like some like I think it was like rats or something, and I was convinced that they were part of the Russian. I don't know. But then you wake up and you're like, oh, I'm just crazy. That's all that's happening. Anyways, I thought I'd share that stupid story.
That's you to have that thing where you're like falling asleep and then you start feeling like you're falling.
Yes, so that's part of it. It's just, you're having sleep effects when you're kind of still awake still. And actually, I think that, because, if you get into deep sleep, you are paralyzed, like as part of the sleep process, you can't move. And that's what sleep paralysis is. You get that whenever you're not asleep. So it's the same. It's the same as all that stuff. It's, I'm saying normal. You're not really supposed to be having that all the time, but there's reasons for it. It's not demons. I'll say it that way. But anyways, sorry to pick up your time talking about bullshit. We've got the sign-in sheet floating around somewhere. Be sure, here it is. I'll put it up here so guys don't forget to sign in before you go. It makes my life a lot easier. Go ahead and get you started, Lauren. Do you say your name Lauren or do you say it Lauren?
Lauren.
Lauren. Okay. Yeah. So when I was a kid, my mom called me Lauren, and so I always expected everybody to call me Lauren. And when people call me Lauren, I'm always like, because I always hear my mom like, Lauren, like in the background, Lauren, what'd you do? It's always me getting in trouble and not my sister.
Some people, they like just saw my name and they're like reading it off of like a roll sheet or something. They call me Lauren, but no.
Yeah, most people call me Lauren. I don't really pick up on it too much, but I always hear my mom's voice, Lauren, with the ah instead of the O sound. I don't know. What is your name? Anyways, you can go ahead and get started. I'll grab mine so I can read you.
Okay, I am presenting on leprosy.
Hopefully, if you want to just click on the slide, and then you can use the right and left arrows after. Hopefully. Yeah.
Okay, leprosy is a bacterial infection and it affects the nerves, skin, mucous membranes, and eyes. If it's left untreated, it can cause paralysis and nerve damage, and this leads to numbness and disfigurement, especially in the face, and then you have a loss of fingers and toes. It's also known as Hansen's disease, and it is caused by Mycobacterium leprae and Mycobacterium lepromatosis. It is a gram-positive bacteria. It's acid-fast with hydrochloric acid. It's rod-shaped, and it's usually seen in single cells, but occasionally in pairs. And it typically exists in clusters in infected cells. So the signs and symptoms from the skin include nodules, thick, stiff, or dry skin, swelling and lumps of the face and earlobes, loss of eyebrows and eyelashes. Signs and symptoms of nerve damage would be numbness, muscle weakness, paralysis, enlarged nerves, and eye problems that could eventually lead to blindness. And then the nasal symptoms would include like a stuffy nose or a bloody nose. And then leprosy is often mistaken for tuberculosis, syphilis, and psoriasis. And something crazy is you can be infected for up to 20 years before you start to show signs or symptoms of it. Mode of transmission is from person to person, typically, so from droplets from a cough or a sneeze. And then it can also be contracted from armadillos, but that's not as likely as human-human transmission. Risk factors would be the further south you live, the more at risk you are, because the further south, the more armadillos are infected with leprosy. So like there have been some cases in Florida. And then of course spending time with an infected person for extended period of time. Weak immune system, living or traveling in endemic areas like parts of Asia, Africa, and Latin America, and then poor living conditions. Virulence factors. So leprosy contains the mycolic acid, which is specific to mycobacteria, and it has the lipid-rich cell wall. So this makes it harder for the immune system to recognize and respond to that bacteria. And then it's very slow-growing bacteria, so since it's not rapidly multiplying, your immune system isn't alarmed by it, and so it doesn't recognize it as easily. And then leprosy uses PGL1, which blocks certain cytokines, and these are crucial for activating macrophages, which those allow for leprosy to bind to Schwann cells, which are the cells in your nerves. So that's why it can link to a bunch of nerve damage. And then leprosy loves cooler temperatures, so it hangs out in like mucous membranes, ears, and skin. Diagnostic methods would be skin smears or biopsies to detect from skin lesions or tissue, acid fast staining to look for that mycolic acid, PCR, and ELISA to look for the different antigens or antibodies. Care and treatment would be, it's completely curable, and most people are actually immune to it now. But if you do contract it, it's a multi-drug therapy of antibiotics for like 6 months to a year. But they have developed the BCG vaccine, and this is, highly recommended where leprosy is common. And then, of course, if you've contracted it, you should keep a close watch of your hands, feet, and eyes. And some interesting history. So back in the 20s, people who contracted leprosy were actually sent to leper colonies for complete isolation to prevent the spread to other people. And then historically, so in the Old Testament, like the Bible, leprosy is Hebrew translation means general uncleanliness, which could cause many skin conditions. So the leprosy that is seen in the Bible is actually not what we see today. That's all.
I feel like leprosy is one of those things we've all heard of, but we just don't know like really a lot about it. All right, Angie. Should be able to let's see, make sure it works. Yes. So right and left arrows.
Okay. Yes. So I will be presenting elephantiasis. So what is elephantiasis? So basically, it's also known as the lymphatic filariasis. It's a skin condition disease that causes severe inflammation and it swells up all parts of your body depending where you get it, which we'll get into that in a bit. So this also thickens your skin. So it causes like inflammation in your skin. Honestly, like it could happen like in your arms and your legs, even your genitals or your breasts, which is kind of crazy. Also on top of that, it also thickens your skin to make it feel like elephant skin, which is where the elephantiasis word comes from. So this disease is also transmitted by mosquitoes. Crazy, right? So the mosquitoes inject like scleral parasites, which is like obviously more common in the subtropical and tropical area. Okay, some signs and symptoms. So you're basically asymptomatic. Most of the time, you won't really see any signs until like after years or like even months also. Um, but either way, you're asymptomatic, like it's still working in the inside, so it can cause damage internally. And some differential diagnoses will be cellulitis, chronic lymphedema, and then um, lymphitis. Some treatments, so we would use anti-parasitic antibiotics, and then in some cases, chemotherapy. So there's actually not really a cure for elephantiasis, neither are vaccines. So basically the antiparasitic antibiotics would just like basically kind of help calm down with the symptoms and stuff. But some examples for that would be the albendazole and then the ivermectin for those medications. And then for the patient care would be like kind of like addressing the long-term disability of this, obviously because of the swelling could cause like you not being able to walk later. Also the mosquito control would be a big one. Okay, so the pathogens for elephantiasis are the parasitic nematodes, which are like the long threaded like worms as you can see on the picture. In some cases, which are called Listeria bandicrofti, and then Bergia malayi, and Bergia timori. And their taxonomy families is I probably won't attempt to say that because it wouldn't embarrass me. And then the genome is a small and compact, and their life cycle includes human hosts and mosquito vectors as well. Okay, so what helps it do its job? So basically, it involves using proteins to modify the host and immune response, helping the pathogens avoid detection. Eventually, the little worms will become big worms, and they will colonize lymphatic tissue by living in the lymphatic vessels, leading to inflammation and tissue damage. And the microflare are released into the bloodstream, which causes the sustained of the infection, which keeps it going. Okay, some risk factors include the tropical regions and of course poor sanitation and limited access to healthcare, and then also like the frequent mosquito bites, of course, and then also long term could probably cause a weakened immune system, which makes you more prone to get any like severe diseases later. Okay, so diagnostic methods. We would use blood smear microscopy, and ELISA, and then PCR to detect the elephantiasis. And then fun fact, about 120 million people worldwide and 1.5% of the population have elephantiasis, which would be more in the South America, Asia, and Africa places. Also, elephantiasis is very rare, not care. I misspoke that. But here in North America, you really won't see that here because that parasite that causes that stuff, like the mosquitoes here don't really carry it, which is why it's not like that be. But yeah. Thank you.
All right, I'm just pulling them up, guys. So when yours comes up, feel free to come up here. Hopefully this will behave. Yes, okay, all right. Just, you can use the arrows or clicking.
Okay. All right, so we're gonna talk about the smallpox virus. It's highly contagious and deadly disease that is caused by the variola virus, which we'll talk about later. It started, well, let me say, this has been around for over 3,000 years, but it came here in the 17th century. In 1796, Edward Jenner created the first vaccine for it that was successful ever. But it was introduced in America in the 1800s by Benjamin Waterhouse. Um, 300 to 500 million people have been killed by this virus, and the fatality rate is 30%. The last known case of this virus is in 1978, but the virus has been eradicated in 1980. You can get it from droplets, airborne, and direct contact. It is family to the Poxviridae virus and genus to the orthopox virus, which has 11 other species in it, which include, I'm sorry, guys, the keypox virus, the cowpox virus, and the vaccinia virus. It is one of the largest and most complex viruses. It is double-stranded DNA. It is brick-shaped. It is modal, I'm sorry. And it is made up of 90% protein, 5% lipids, and 3% of DNA. It replicates in the cytoplasm and steals the envelope from the host. Common symptoms and signs, which are familiar: fever, fatigue, chills, vomiting, rash, blisters, bumps, scabs. It takes up to 7 to 17 days to even show any signs or symptoms. At 12 to 14 days, you will get a rash all over your body, which it will last for 24 hours. And it'll fill up with pus and bust, and it's just nasty. If you can see, it looks like the mother is trying to get a little bit of it too, but that poor baby is got the most of it. Once you have developed a fever of 101 to 104, you do have it, and you will have it until the last scab falls off. It can be mistaken for chickenpox, impetigo, scabies, and hand, foot, and mouth disease, which are all pictured here. And anybody can get it, anyone. If you're unvaccinated, if your immune system is weak, and if you're pregnant, those would be the ones you would get it the most. Okay, diagnosing. would be a PCR, viral culture, skin testing, electron microscopy, and serologic testing. But the best one is gonna be the PCR, just because it has that real-time mechanism. Treatment, there is no treatment. If you get through it, you get through it. If you don't, you don't. But being vaccinated is gonna be the best way to prevent it. When you do have it though, just make sure you isolate yourself, have plenty of fluids, medication to help your symptoms. And I think that's it. Yes.
All right, you should be good to click or space bar arrows.
Okay, so I will be presenting on feline calicivirus. So feline calicivirus is a viral pathogen that is common in cats. It was first discovered in the 1950s, and it primarily causes respiratory fevers and respiratory symptoms and fevers, but in severe cases, it can spread throughout the whole cat's body and cause a systemic infection. So, feline calicivirus is caused by a virus. It belongs in the Caliciviridae family in the virus genus. It has a non-envelope icosahedral capsid, and so it has the ability to persist in your environment, allowing it to survive on surfaces for an extended period, and this increases the chances of spread between cats. And so it can last up to like 7 to 21 days, but in severe cases, it can last up to 1 to 3 months. But it does eventually go away on its own. And it is very contagious, but it can be deadly too. And so some virulence factors of feline calicivirus includes viral capsid protein. So this adds protection to the virus and allows it to attach and enter whole cells. It also shows antigenic variation, meaning that it avoids detection by the immune system. And it has cell tropism, meaning that it targets specific cells that causes respiratory issues and eye discharge and oral ulcers. So it can be transmitted through direct contact with other infected cats. It can also be transmitted through contaminated surfaces and objects such as the food bowls, their litter boxes, the floor, or whatever the cat touches. And then aerosol transmission can also occur when the cat sneezes. And so to prevent your cat from catching the virus, you should isolate the infected cat from other cats. You should clean and disinfect any contaminated objects or surfaces. And you should make sure to vaccinate your cat to prevent it from getting any of the severe infections from the virus. And some risk factors of feline calicivirus includes kittens and unvaccinated cats. And so they are at high risk due to their weak immune system. And then cats that are in multi-cat environments can also be higher risk due to being in close contact with other cats that can probably have a virus. And stress and poor hygiene can make the cat's immune system weak, and so it can make them more susceptible to the disease. Some signs of feline calicivirus include sneezing, nasal discharge, drooling, fever, loss of appetite, oral ulcers. And so sometimes these signs can be mistaken for other diagnoses, such as feline herpes virus, where they can have similar respiratory symptoms, and feline leukemia, where they both can develop oral ulcers in the mouth.
So to test if your cat has feline calcivirus, you can collect, well, samples are collected from the nose, the eye, or from the mouth, and then they are sent to the lab for a polymerase chain reaction test. And then this test confirms for the presence of the virus. And then immunochemical, immunohistemic, I don't know how to pronounce it. Immunohistochemical test involves applying antibodies to the tissue samples, and this is particularly helpful to detect viruses in the sores or infected organs. And to treat a cat with feline calcivirus, you should give some anti-inflammatory drugs such as meloxicam for the mouth pain and fevers, and make sure it stays hydrated, and give it some strong smelly foods to encourage eating. And there are also eye medication, nasal vaccines, and core vaccines such as Calicivax. So Calicivax can just reduce some of the severe infections from the virus, but it is not a cure. Yeah, and there's no cure for the virus either. So yeah, you just have to treat it. That's all. Thank you.
So you can use the arrow, spacebar, or click to advance your slide.
So I did. Bacteria was pertussis. And pertussis is a serious respiratory infection that's caused by the bacterium Bordetella pertussis. It's going to be a fastidious gram-negative bacteria. It's going to produce multiple antigenic and biological active products. Pertussis is also called whooping cough because it causes this severe cough, these violent coughing spells. And so this cough is so intense that when you start coughing, it causes all the air to be expelled out of the lungs. And when you're forced to take a breath, inhale, it causes this whooping sound. So that's why it's called whooping cough. Pertussis affects humans only. There's no vector, animal, or insect sources. It's going to affect all people of all ages, but it's going to be very important and dangerous for infants and those with 1 year of age because it can cause significant death in that group. Those that are 2 months of age and younger are very vulnerable because they cannot start the DTaP series for vaccination until 2 months. That's when they can start it. So transmission, pertussis is very highly contagious, and it's going to be spread from the respiratory droplets that are suspended into the air. Pertussis bacteria, it's going to attach to the cilia in the upper respiratory system, and there it's going to start to release toxins, which is going to damage the cilia and cause the airway to swell. When the airway swells, this is going to block the flow of air into the lungs. The lungs can actually collapse, and it's going to be considered to be able to be passed for up to 3 weeks. When coughing starts, I'm sorry. So virulence factors for pertussis are going to be adhesions, and these adhesions are going to be this filamentous, hemagglutinin, fimbriae, and pertactin. And it can bind, that's what adhesions do, they bind to that ciliated epithelium. And you can see here where these are like healthy cilia, and this is where those adhesions have caused this bacteria to start sticking there. Tracheocytotoxin is going to be another virulence factor. It's a protein that causes ciliostasis. So, the cilia is supposed to help
lungs, pathogens and all that, and be expelled and moved. So this cytotoxin stops that. It causes the cilia just to stop. So therefore, things get trapped in there. You can't get the mucus out. It's gonna plug the airway up. You can't breathe. This adenylate cyclase, it's another major virulence factor. It's gonna cause that bacteria to compound colonize there. It's also gonna stop that immune response that's there in the lungs. And pertussin also has a pertussin toxin, that's a virulence factor, and it's also gonna cause this colonization of the pertussis there in the lungs. So symptoms during the first couple of weeks, 1 to 2 weeks, it's just gonna be just like a common cold. The same stuff like slight fever, mild coughing, runny nose, apnea in babies, that's just where they pause, they stop breathing. Usually after the first 1 to 2 weeks, that is when that violent coughing is gonna start. That whooping sound that's notable, you're gonna have vomiting, difficulty breathing, sleep just due to exhaustion from coughing. Rib fractures can happen, exhaustion, those are the later symptoms. Serious complications are gonna be pneumonia, pulmonary hypertension, dehydration, subdural hematoma, encephalopathy, seizures, rib fracture, apnea, and death. Because you gotta think the blood and everything is going to the lungs to get oxygenated. So when you clog that factor up, it's gonna affect other things as well. I don't know if my slide will play this YouTube, so you can kind of hear how it sounds. Will it play?
It's probably not anything. Let me, just a quick. It's because it's not my slide. I went to the computer and like, I don't know what you're trying to do with somebody else's crap here. So I have to like, yeah, enable the sharing here.
Okay.
It's just. Shouldn't be a big issue. What am I not seeing? There it is. Okay. So it should now, let's see if it'll do it. Enable content. There we go. Okay, now it will do it. There you go. That's terrible.
Video that came from, and it is up like an actual little infant, little newborn baby that's coughing in that. And it goes on for like.
Yeah,
5
that's like a
minutes.
just
Oh
a
my
10-second
God.
clip, but
Yeah,
yeah, it
that
just
is
usually
like-
starts.
Yeah, they have to cough like that, and they don't stop. It's that, is it, for like all the time.
Yeah. So how are we gonna test? There's 3 ways to test for pertussis: culture, PCR, and serology. And culture is gonna be considered the gold standard. That's just gonna be the most specific way that we can test for pertussis. It's actually collected on a nasopharyngeal swab and collected on this charcoal medium that you see present. And it's gotta be collected really between the first 2 weeks. That's gonna be the best time to test it because sensitivity decreases after the first 2 weeks. And the disadvantage for this, even though it's the best way to test, is that it takes so long. It can take a week or longer, some say about 2 weeks to get the results back. So that means you're just gonna delay treatment for that person if you're waiting on this culture to come back. So you wanna use it, but you don't want it to define diagnosis to start treatment. PCR is gonna be the most rapid test that we have available. Again, that's gonna be collected on that nasopharyngeal swab as well. It's gonna be great for sensitivity, but it's gonna vary in specificity because we're gonna get more false positives with this one. You can do this for up to 3-4 weeks. After cough onset, after the 4th week, the amount of this bacterial DNA is decreased in the nasopharynx. And if you can see, I don't know if anybody's been tested for COVID in this room, this is a real thing. Getting back there in the base of that nasopharynx is very uncomfortable. I mean, I have been a nurse for many years, and when COVID came and we had to start doing these, I got bit, I got spit on. I mean, we had to restrain people because you have to get in there and actually swab that and then rotate it for about 20 to 30 seconds. And it is very uncomfortable for you. Then you get somebody with a deviated septum, and it's even worse. So this is, as a nurse, you will be doing this all the time. Nobody's gonna do that for you. You will take the abuse. Serology testing can be done also where they test for antibodies, but it's not gonna be, the CDC says that there's several assays that are gonna vary on whether these tests are accurate or not, but it is still another way. And it's supposed to be, you can detect the antibodies for up to 8 weeks using this testing method. How do we treat pertussis? So we treat it with antibiotics, and we want to start treatment early. That's why we want to get that PCR so we can know early and how to start treating it. We're going to treat it with antibiotics. Macrolides or mycins are going to be our drug of choice. Preferred treatment for babies is going to be azithromycin. If you're allergic to the macrolides, then you do have the option of that sulfonamide antibiotic, which is going to be that trimethoprim sulfamethoxazole. And there really is no treatment for the cough. If you get it, those cough suppressants, albuterol, antihistamines, corticosteroids, they're not really proven to really help that cough. I mean, there's not been really anything that can help that. But however, if you start antibiotic treatment early, it can help lessen the severity of it. You're gonna just wanna manage your symptoms with rest. You wanna stay hydrated, prevent that dehydration, cool mist humidifier to soothe the lungs and to loosen mucus in the airway. Keeping your environment free from irritants and dust and smoke, things that just can start making you cough. You want to keep your babies' air passages clear. That suction bulb that babies hate is your best friend. Keeping their pathways clear, monitoring their breathing, because sometimes you may need some supplemental oxygen just due to that respiratory being plugged up. Pertussis, it can live on our surfaces for 3 to 5 days. So disinfecting is going to be key. You're going to want to use low concentrations of chlorine and alcohol to do that. And prevention, the best way is going to be vaccination. You want to start that series in your babies. It's 5 doses. You're going to start it at 2 months, 4 months, 6 months. They'll get another one between 15 to 18 months of age, and then between 4 to 6 years old. Adults should get a booster when they're between the ages of 19 to 64. Expectant mothers are going to want to get one in that 3rd trimester of pregnancy, and anyone that is going to be around your baby should get a booster when they're going to be around infant children. So people who have been vaccinated can still get pertussis, but the illness is generally milder than those who have not been vaccinated. Pertussis vaccination has significantly dropped the number of cases in the United States. Before vaccination, we could see about 200,000 cases of pertussis a year, and about 9,000 of those would be deaths. But since vaccination, we only see about 20,000 cases, 10 to 20 is variable, and usually about 15 deaths in the United States. And this is totally preventable if we vaccinate. The end.
All right, that's my turn now, right? It's a lot. Okay. All right, so we know about our respiratory tract. Hopefully I don't have to review this too much. We are aware of parts of our body involved. Some of the language that I might be using, like pharynx, that's referring to your throat. So we will talk about that a little bit, but we know some of our defenses that we have, our physical defenses. We have the cilia that are like inside of your true respiratory tract down in your lungs. And that helps move mucus and debris out. And then the coughing helps to expel that. And then you either cough it out or you swallow it to get rid of any microbes that could be in there. The hairs of your nose also help trap debris to prevent any infection that way. For our second and third line defenses, we have complement action in the lungs. We have cytokines and antimicrobial peptides in the mucosa in those areas. Macrophages that are located in the alveoli called alveolar macrophages. And then secretory IgA is that antibody that's associated with mucosa. So it works better in the mucosa than like IgG does in your bloodstream. Yes, so our normal biota, we do have quite a good number of other microbes that can be found in your respiratory tract as a whole. What you see in your sinuses and stuff and in your throat is different, of course, than what you see in your lungs. But we can have colonization with bacteria and fungi, and even protozoa can be part of your biome here. These are our normal biota, but these guys can also be associated with disease if they overgrow or get transmitted to other areas of the body they don't belong. Streptococcus pyogenes, that's strep throat bacteria, but you can harbor it naturally. Haemophilus influenzae, Streptococcus pneumoniae, which we learned as the most common cause of communally acquired meningitis. It's also going to be the same for the pneumonia. Neisseria meningitidis, the worst kind of bacterial meningitis that you can get. You can harbor this in your sinuses as part of your biota. And of course, staph, which is like anywhere. So based on whether or not you carry these guys, you're going to be more susceptible to the diseases that they cause, but you can also be a carrier and transmit it to other people. We'll start with the common cold. This is caused by over 200 viruses that can cause the cold. And that's crazy to think about, but so you get diagnosed with the cold, this is a syndrome, right? So we're talking about symptoms associated with having a cold versus what can actually be causing the cold. It could be any number of viruses. You're usually not going to have a fever associated with this, but it is possible to have a low fever with a cold. Basically, very similar symptoms to influenza, usually not a fever and usually not as much pain associated with a cold. And then the difference, of course, is going to be what's causing it. So RSV is also another potential cause of those cold-like symptoms that we might diagnose as a cold, but your causative agent could be RSV. If you're an otherwise healthy adult, that might be the case for you. If you're a child, we're going to be more concerned about whether it actually is RSV because it can be more dangerous. Sinusitis, itis being inflammation, sinuses. So this is going to occur, it can occur separately from like allergies and then secondary infections, or it can be part of other illnesses. So we can have this occurring along with other things. Caused by viruses, bacteria, and fungi, but actually more commonly it's going to be viruses. So keep that in mind as well. But secondary infections from bacteria can happen as well. Notice we have otitis media. And otitis media is the ear infection. It's also often associated with like a secondary aspect of another illness. You get buildup of the fluid in the ear, and then it can migrate to other aspects of the respiratory system and other parts of the ear. Secreted fluid is called effusion. It can build up, cause a lot of pressure. Bacteria can grow in there. It's not a closed system, right? So you know your eustachian tubes that connect your throat to your ears. If those get too inflamed, then we have the backup of that fluid and the bacteria can't drain out properly, and then you get development of the middle ear infections. You have a feeling of pain or fullness in the ear. You might have loss of hearing as a result of the inflammation in the ear area. Young children will often be irritable, have trouble sleeping or eating or even hearing as a result of this, and they can't really necessarily tell you what's going on as well. So that can be a problem. It can cause your eardrum to burst, but you can implant tubes to help drain the fluid to prevent permanent damage from it. So typical recommendation is to wait 72 hours. If it's not improving, then go to the doctor and see if you need to get any antibiotics or other treatments. It's usually caused by strep pneumoniae. So that's the one that's the common meningitis and pneumonia. So this guy is little fingers in a lot of areas. It can also be caused by Candida auris, that fungal infection. So auris, think of like, being able to hear things. So I don't know how else to say the auditory, right? So That's how I remember that one, but this is not communicable. You don't get it from other people. It's usually something that progresses from other things going on in your body, usually from the eustachian tubes, the drainage tubes being overinflamed. Next is pharyngitis. So this is inflammation of the throat, often caused by strep pyogenes, so strep throat, or Fusobacterium necrophorum. So this is also Fusobacterium species associated with trench mouth, which we'll learn about in the GI chapter when we get back from Thanksgiving. But it's pretty, these guys are pretty ubiquitous as far as not everybody might harbor these, but you can get them from other people who do carry them, and it's not uncommon to see them. You'll have white packets on the walls of the throat, difficulty swallowing, foul breath, sore throat. And if this is a viral pharyngitis caused not by bacteria, you still have mild symptoms and hoarseness, but it's usually not as bad or not going to affect you as much as the strep throat will, but still it's pharyngitis, just viral or bacterial. Bacterial is usually worse. It usually has fever and that foul breath associated with it. Here's an image of someone's throat with the bacteria. You can kind of see growing on the tonsils in the back of the throat. I used to have tonsils that when I didn't have an infection, they almost touched in the back of my throat. And when I did, y'all, it was like I couldn't even breathe. They were so big. And I got them removed just a couple years ago, finally. And it's like magic. You have no idea what, I didn't know what breathing was like. So now I feel the pain of you folks out there who are still struggling with your tonsils, if you still have them. Yeah, I feel you, believe me. I used to have all sorts of problems with it. So this is strep pyogenes and what it's associated with. It can lead to the development. You get strep throat, you can get scarlet fever, you can get rheumatic fever. So we're having antibodies against the strep and that affecting the heart and inflammation in the heart. And then that acute post-streptococcal glomerulonephritis with those immune complexes that formed that we talked about in the type 3 hypersensitivities. It has capsules and it can form a slime layer as well. So that helps it adhere to tissues and be harder to phagocytose these guys. And it creates certain toxins and enzymes that help it infect tissues, breaking down your like basically your what am I trying to say, extracellular matrix between your cells so it can invade in deeper tissues. Keep in mind that Streptococcus pyogenes, strep throat, scarlet fever, and rheumatic fever, but also things that are related to other diseases that can become much worse, like necrotizing fasciitis and other things. So this bacteria, again, associated with a lot of other diseases. And maintaining, making sure that you're getting treatment for it is important to prevent the worst versions. So again, another bacteria associated with bacterial pharyngitis is Fusobacterium necrophorum. It will cause something called Lemaire's disease, where you get peritonsillar abscess. So around the tonsils, you get these abscesses that form that become so swollen, they can rupture, they can cause tissue damage, and it can be life-threatening if that bacterial infection spreads to into your bloodstream and other parts of the body. So usually we'll drain those with a needle and then put you on antibiotics. We've talked about whooping cough, so I'm not really going to go over it, but you do need to know the 3 different phases of whooping cough and what's going to happen in each phase. So do be aware of that. That's always going to be on the exam. But yeah, catarrhal phase, you have cold symptoms. Paroxysmal phase, whooping cough. Convalescent phase, you're getting over the actual whooping cough aspect, but you've had that ridiculous cough for so long, your cilia are damaged and you're still prone to secondary infections, and it can take months from healing. For healing. This is actually the video that was associated with the sound clip that we got before. This is that little baby from that. I'm not going to play this video, but you've heard it. But this is common in little babies. They haven't been vaccinated. And so when you're an older adult, it's not as serious. You can still get some symptoms, but it's usually not too bad. But if we don't keep up with your pertussis vaccine, then, you know, as a grandparent, you go see the little baby that was just born and you give the newborn baby pertussis and you didn't even know that you had it. So you want to keep up with those vaccinations to prevent the little babies that can't get the vaccinations yet. This can be deadly. I mean, you've heard that baby coughing. That is what it's like constantly for that baby. And they will die potentially from secondary infections is more common, but you can die from asphyxia because these babies don't really need to take a breath. They haven't really learned that yet. So just cough and cough and cough and cough and cough. And you know how you'll cough and you'll be like you heard the baby doing to take a breath. But like if you watch the video, the nurse that is holding the baby, she's like, come on, take a breath, take a breath. But the baby doesn't know what's going on. So that can lead to hypoxia and things like that. That can be actually pretty serious as far as causing problems with brain function and things. So, all right. This is caused by, again, Bordetella pertussis. It's a bacterium and it has these toxins involved. I'm not going to go over this information past that. Okay, RSV is called respiratory syncytial virus. And syncytia are whenever, like, if you're a virus, you have spike proteins that help you get in through the doors of the cells, right? Well, they will put those spikes on the outside of your cells and then bud through it to coat themselves, right? They're on the surface of the cell before that happens. So because you have those spikes to get into other cells, now you have the spikes on cells that try to get into the cells next to them. And so they use the key on the cell to the next cell, and then they merge together. And that creates like mass cells that are all combined into one big old membrane by doing that. And those are syncytia. So that's syncytial. That's a very characteristic aspect of this virus. A lot of viruses do that, but this is just, you can like look at histology slides of this and see that syncytia forming between cells. So what used to be several separate cells will now be like one big old combined cell. You know, cold-like symptoms are a lot more dangerous to younger children under 6 months of age. Can be deadly. So you want to look out for symptoms. Like I said, cold-like symptoms, you can develop croup and trouble breathing. It can be bad for the smaller children, but we do have antiviral treatments for it for severe cases. Next, into flu. This is influenza virus. So RSV virus, influenza virus, cold virus, pertussis bacteria. Strep throat can be strep, or you can have sore throat from viruses. So influenza, this is the flu. Usually is maintained in bird and swine populations. This is considered a zoonosis, so you can maintain it in human populations. The new strains are always coming from the animal populations or mutations within the human populations. We'll talk about the differences there. We know the symptoms. I'm not going to go over them with you guys, but they're pretty characteristic for flu. And you can develop pneumonia usually from secondary infections. And it's usually more serious again with those with lower immune systems, like the young or the old. But if we get new strains, like we have with pandemic versions of the flu, those can affect even healthier people and be quite severe. So this can be a deadly disease, not in a small way. So on the surface of the flu virus, we have H and we have N, and we classify our viruses by what type of H1 through 5, whatever. I don't know how many there are, but there's a lot, or N1 through whatever. So H1N1, H5N1, H1N5, whatever it is. And so it's the type of classification of H or N that you have that will classify your flu as. H is the hemagglutinin. It binds to blood cells and it's our attaching protein, whereas the N, the neuraminidase, it helps get into cells, it helps bud out of cells, and it breaks down the mucus to make it easier for the virus to move around. We have two different ways that the flu can mutate and change to different strains: antigenic drift and antigenic shift. I used to have like a picture on the old slide that I had of this pig. So the idea is antigenic drift. Imagine you're in an ocean and you're floating around on like a life raft and you're just at the whim of the sea and you're drifting around in the current. It's going to be slow moving, right? But if you were able to shift yourself with a paddle or a motor, you make bigger progress. Same thing kind of here, but drift, you're making just random genetic changes. Some of them will be beneficial to the virus, normal mutation stuff. Shift, you have virus H1N1 and virus H5N5, and they come together and then you get H5N1. They can like intermix their gene segments and create brand new huge changes in these viral gene segments. It has 8 segments in its genome, and so you can switch them up however. But usually what happens is you have an animal, a pig, or a bird that sees both of them at the same time. And now we've made a new virus from just mixing and matching the segments. So that's shift. And the way that I used to remember that is one time I was writing it on a note board for my students, and it was like a picture with the pig and how the bird one comes to the pig, and then the pig has the pig one. They make a mix, and then the farmer gets it from the pig. Well, I wrote antigenic shit on it. And so and so I thought of like how pigs are often in a lot of poop, right? So shit. So I thought of pig and then that shit and then shift. And so that's how I remember that the shift is where those big changes are coming from inside of like one animal. Several viruses come in, reassort those choices of gene segments and make a new one. So big changes from those shifts. Okay, so those are where we get the pandemics from. Oh, here's the picture. This is the picture, y'all. That's the pig. So yeah, 2 different strains, the blue and the orange. Pig is with both. It has mostly orange, but 1 blue. But to you, your body's never seen anything like this before. And so you'll react to it. So when we have big changes in our new pandemic version, that's what's happening. Even the little changes can lead to the drift ones, even those can lead to needing new vaccines. So anytime that we have a change that your body hasn't seen it before, you're going to need a new vaccine for it. So this guy can mutate like nobody's business, as well as shift big changes that'll be really detrimental to the human population. Drier air actually facilitates the spread of the virus because your mucosa will dry out and be a little more irritated and make you more susceptible to getting sick. So being out in the cold doesn't mean that you're like getting sick from the cold, right? But you can be out in the cold, have the dry air from it, and then make you more susceptible to other people's illnesses. So that part is true about getting sick from the cold air. That's not entirely a lie. So you prevent this, we already know, same way that you would prevent, you don't want to get sick with the flu, pretend like you are being exposed to COVID. Social distancing, masking, hygiene. Also, we'll have vaccines available for a lot of the more common strains. There's so many strains of flu out there that we can't protect from all of them. And a lot of the time we'll look at strains that are in other countries as predictors of what we'll have, and we don't always get it right. So this is a guessing game, and that's why it's not always going to protect you, but they do the best that they can. We're trying to develop a universal vaccine, but this guy just mutates so much, it's hard to keep up with. There are antivirals like Tamiflu and Relenza that will work against the neuraminidase function. So that will keep the virus from getting into cells and from budding out. But if you don't start that early, then you'll already have the infection to where it can't control it as well. So you want to take that early on in symptoms. Next is tuberculosis. This is a bacterial disease. We know this one, y'all. If I can't shove it down your throat, it's enough. It's Mycobacterium tuberculosis. So this is an acid-fast bacterium. It has that mycolic acid on the outside, makes it hardy, resistant, hard to clean. It's resistant to drying out and things like that. In fact, you can get micro aerosols that can persist in the air for like 8 hours or something like that. So somebody coughs in here now, 8 hours later, somebody walks in here, they can get your tuberculosis. So it doesn't require very many bacteria to start the disease, only about 10, which is pretty low for bacteria. So it's pretty easily transmitted, not as much as like measles or something, but still. It's a re-emerging disease in society because of the HIV epidemic. You guys, I didn't give you the statistic, but if you didn't know, over 50% of the population of adults who are in the sexually active ages of their lives, those adults, over 50% of them in South Africa have HIV. Over 50% of them. Imagine that. HIV, that's forever. So there's a high incidence of tuberculosis amongst populations that have HIV. And this is a problem here in the United States with our homeless populations in particular. So they'll have usually HIV and tuberculosis, if not one or the other. We also have drug-resistant strains. This has been a really big issue in Russia, where they were not completing their regimens for their antibiotics, and now we have a severe increase in the resistance amongst tuberculosis in those areas. And about a third of the world's population right now has tuberculosis. Like they're infected, they're latent with it, or they have symptoms. A third of the world. So we have primary and secondary or reactivated tuberculosis. The primary tuberculosis is your initial infection. You get the bacteria, your body walls it off, and these structures called granulomas or white blood cells try to contain it. Your macrophages in your alveoli, your alveolar macrophages, comes in, sees that we have PAMPs all around. We're going to start activating your immune response. It's great. We're going to phagocytose these guys and get rid of them, right? Except that these guys want to live in those cells. That's where they live. So you go phagocytose them, and they're like, thanks for the nice, new apartment. So they just hang out there for a long time. You might have some inflammation and irritation in the primary, but usually you can be asymptomatic with your primary tuberculosis. However, if I look at your lungs in an x-ray, you will see where these granulomas vesicles have formed. They can become quite large, become caseous. So that means they are they look like cheese. Gross. Think about the bacteria that's there, and it has that consistency. It's kind of hard, and those can calcify and become literally hard. So we can test you, the skin test. We've talked about this, so I'm not going to review it too much, but if you have seen tuberculosis, your PPD skin test will get inflamed because those T cells will have seen it before and have that response to it from being the delayed type hypersensitivity. The secondary or reactivation tuberculosis is where you have the symptoms coming up as tuberculosis. So you can harbor tuberculosis for years and not have any symptoms, but once there's some reason, low immune system, you're stressed, whatever it is, you can have reactivation. If you leave tuberculosis untreated, especially once it begins into the reactivation phase, it's a 60% mortality rate, which is crazy. We're talking 6 to 9 months of antibiotic treatment. So good luck. Most of us aren't going to adhere to that. So it's easy to imagine why we have resistance developing. When you get into reactivation tuberculosis, this is where we think of tuberculosis proper. You have violent coughing, greenish and or bloody sputum, low-grade fever, anorexia. This just means you don't want to eat, so you're not eating. Weight loss, fatigue, night sweats, and chest pain. So this is consumption. That's why they call it consumption. You are wasting away and coughing up blood. We already talked about it being acid fast. There's another version of it caused by Mycobacterium avium. It still causes tuberculosis as like the symptoms, right? But the bacteria is just a different species, but we only see this one associated with HIV. Just some pictures of it. You guys know you better know what an acid fast looks like, right? Acid fast, the negative cells are blue, the positive are reddish pink. So carpal fusion, right? You think that's going to be on your test? Yeah. So tuberculosis transmission, these are the things that are going to make your risk factors. You're having inadequate nutrition, debilitated immune system, poor access to medical care, lung damage, and then just genetic susceptibilities involved with this, okay? It just occurred to me something that's going to be weird about the test, of course. So anyways, moving on. So we see this in like sub-Saharan Africa, where we have malnutrition and we have spread of this bacteria, and it can disseminate into other tissues of the body, like into the gut area. You get a waxiness that's over the organs and things like that. So the skin test is the Manto test. We have the antibody methods of recognizing these guys and PCR too, as well as X-ray. With the skin test, all of these can be used to identify. Usually we'll do a sputum collection and then do the acid fast test on that and then move from there to confirm the diagnosis. There are antibiotics available that you don't necessarily need to know what they are, but they are there. We're just having more resistance, and we're talking again 6 to 9 months of treatment. You'll start with all a series of the antibiotics, and whichever one seems to have improved your symptoms the most, then we'll stick with that for the rest of the treatment for the 9 months. Pneumonia, this is a syndrome, an anatomical syndrome. So we're basing this on signs and symptoms, and it can be caused by a ton of different things, bacterial, viral, fungal, all sorts of stuff. So very, very common. We know this is, we're gonna have fluid collecting in the alveoli. We're not able to clear it out effectively with our coughing, so it will just settle down in there. Sometimes that can be caused by our cilia being damaged from the coughing. And this can be a secondary to something else, or it can be the actual infection that you have as well. So usually have some sort of cold-like symptoms, then it will move into the lungs with the chest pain, fever, cough, and sputum. You look pale because you're not getting enough oxygen. You'll have difficulty breathing as well. How bad it is depends on what's causing it. So this is just an image of that fluid building up in the alveoli with the bacteria and all that building up down in the deep parts of your lung, and you can't clear it out as well. The first one is the most common one. This is Streptococcus pneumoniae, most common form of meningitis, bacterial meningitis, most common form of communally acquired pneumonia as well. So usually about 50% of people can carry this. It's just part of their normal biota, but it's just a matter of where it's supposed to be in your body and how much of it is supposed to be there. So it shouldn't be down in your lungs, and it shouldn't be in very high numbers anywhere in your body. You can share this with other people. This one's alpha hemolytic. So you see that greenish tinge around it. That's what we're talking about with the alpha. It does have capsules. We talked about strep pneumonia before, so I'm not really going to go too much into it, but it does have capsules. It blocks complement, and we can treat this with certain antibiotics. Next is mycoplasma pneumonia. So those ones are the guys that don't have a cell wall, which makes them pleomorphic. They don't have a set shape either. This leads to mostly what we call walking pneumonia. You have pneumonia, but you can still function on daily life, and it is what we call an atypical pneumonia. Legionella pneumophila is another bacterial infection associated with Legionnaire's disease, or we saw it in a 1976 outbreak in a like American Legion convention in a big old building, and all these old guys were there for their convention, and a lot of them ended up with this disease. And it was traced back to this bacteria that was found in like the cooling towers for the air conditioning system building. So sick building syndrome, that this is one of the causes. Well, you can get respiratory diseases from, your air supply from buildings. Legionnaire's disease. All right, then this is Histoplasma capsulatum. So this is histoplasmosis, and it's going to be very similar presentation to tuberculosis. This is a fungal one, most commonly associated with AIDS, but anybody can get this if you have a weakened immune system. Next is pneumocystis gyrovecki, also associated with AIDS. This is a fungus that is pneumocystitis, that's going to be associated with the drugs. This just occurred to me that this one is the protozoa. This is a protozoa. That's my fault. It doesn't say it on here, but it is a protozoa. Protozoa, histoplasma, okay? Pneumocystis gyrovecki is the fungus. Why doesn't it say that on there? Probably says it on this freaking thing. It doesn't, does it? It's also fungal. I'm wrong about both of that. All of it's fungal or bacterial, okay? There is a protozoal one. I just can't remember which one it was. It's not on here. You don't have to worry about it. It makes that a lot easier. Well, obviously, you can tell that's not going to be on the test. So I'm not going to ask you about if those were fungal or not. But if they're a cause of pneumonia, that could be a question. All right, so 30 to 50% mortality rate for healthcare associated pneumonias. So you get put on a respirator, you're in a hospital, you're debilitated, you're not helping circulate your lymph by moving around, you're more likely to get these infections, usually caused by a combination of bacteria, and they're usually just common in the environment, and they move into your lungs because of you being, basically immobilized and things like that. So it's pretty deadly as far as a concern there. So about 1% of hospitalized people can get it. If you get it, 50% mortality rate, which is crazy to think about. So you want to be sure you're on top of taking proper care of your intubated or patients that have tracheostomies and stuff like that. Machines are breathing for them. Keep the machines clean, keep them clean, keep everything nice and clean. And what do I say? Elevating the head, encouraging coughing if they are conscious, deep breathing, and ventilation and cleaning. Next, moving on to hantavirus pulmonary syndrome. It's a viral infection. We have mice and things like that will be rodents, usually in the southwest of the United States. They leave behind their feces and their urine. It dries up. You sweep it. It gets in the air and you breathe it in. You can get hantavirus pulmonary disease, and that can progress to a version that can be hemorrhagic in nature. This is a pretty serious one. We have a mortality rate of about 33% in everybody if you get it. So that can be pretty bad. I'm trying to think. Okay, so you get your cold and flu-like symptoms, and then that can progress to the actual pulmonary syndrome becoming severe, where you're having respiratory distress and spreading through the blood, difficulty breathing. It is airborne if you are dealing with the feces and the urine that are being cleaned up in a house, swept, and everything like that. Other than that, it's not really too contagious or anything. We'll look at this with PCR and stuff. These are the cases that we've had in the United States here of hantavirus, and we do have it here in Oklahoma, so our rodents can carry this. It's just more common in the Southwest. All right, it's considered a category C bioterrorism agent, so not as serious as A. So these were our respiratory diseases for this chapter. I'm going to go through the questions. I'm not going to ask them. So what disease is Fusobacterium nephroforum associated with? This is our, for this chapter, going to be the throat, the pharyngitis. There's the word. You guys get multiple choice. All right, what organism causes whooping cough? This was Bordetella pertussis. It's a bacterium. Explain antigenic shift in influenza virus. Blue virus, orange virus, we get some segments from the blue, some segments from the orange to recombine to make a new one. in an animal, and then it gets transmitted to people. What type of organism causes tuberculosis? Bacteria. This is actually a commonly missed question on the exam. What is the most common cause of community-acquired pneumonia? Streptococcus pneumoniae. It's bacteria. Also causes the most common cause of bacterial meningitis. So when we go up to the lab, you guys kind of know what to expect. This setup is completely different. I have to share that lab spot with biomanufacturing now. So I had to leave one of the benches completely open for them to use. So I would have to reset up everything every time. And so that they come in in the afternoon around 4 so that they have their space to work. And then we have 4 students, but still. So there's one bench, it's bench 2, so the one up in the center that is completely empty. We will not be going to that bench. You guys will hand in your scratch paper and your scantrons at that bench. Okay, we'll create a pile there. But we will have, I'll show you guys where everything's set up. We'll start at question 1 in a different place than we usually start. It doesn't really matter as long as you're following the numbers and the arrows to keep up with where you're putting your answers. Double check that you're marking the answer that you're choosing. Right, so if you are like looking at, oh, my answer is C for increases pH, make sure you're actually marking C, because we do have a few people in the last exam that like just didn't double check that stuff and then got it wrong, even though they knew the right answer. They just marked the wrong bubble. That's the fallacy with the Scantron. I do not have very many extra pencils, so I hope that you guys have yours. People have been taking